looked at more molecular recognition part, they're feeding those bacteria, cultivating those bacteria inside the gut. Really a beautiful, elegant symbiotic system, very mutualistic.
So those bacteria they're not behaving like pathogens. They're not trying to camouflage or hide or get away in order to avoid detection by the immune system. They are behaving as if they should be there.
Precisely. Yeah, precisely. It's like they're, the microbiota is an organ system, right? So it's, it makes sense that everything is working together in a cohesive fashion. So yeah, that's really what the immune system is doing right there, is it's interacting with those bacteria. In its interactions with those bacteria, it learns a lot about what kinds of things are dangerous, what kind of things are not dangerous. It's a training, part of the training of the immune system.
So can you talk a little bit about, so when you have bacteria like this that are not pathogenic, that are supposed to be there, contained within this niche of the gut, how is the immune system feeding them, and how is it detecting and interacting with them so that it's keeping them at bay but not killing them or trying to destroy them like it would a pathogen?
Yeah, so in a way it kind of keeps them at bay, but so the way it does it is by it helps them form these things called biofilms. So the feeding process is more or less through mucus. So they digest mucus, we feed them mucus, and there were some really elegant studies in mice that only have one bacteria. You can show if the bacteria can't eat the mucus, then the body, and these are mice, they'll stop producing the mucus that can't be digested by the bacteria, and they'll produce other mucus that can be digested by the bacteria. So mucus is really a good way to feed your bacteria, a nice carbohydrate meal. Of course, we're supposed to be feeding them fiber, and then we do a poor job of that in Western society, but so they have a really protected space. But the molecular recognition part that we were working on is also really elegant. We put out these molecules, which in antibodies primarily, immunoglobulin A primarily, and it helps them form a matrix. So that normally bacteria on their own have to form their own matrix to sort of glom together, form a film, and the films stick much better than a single bacteria. So that's what bacteria do. They grow, it's called the sessile state versus a planktonic state. So that helps in a flowing system. If you look at, say, a sewer pipe, there will be a bacterial lining on those pipes, but the bacteria have to make all their own molecules, whereas we help them by helping them make their matrix.
I see. So these biofilms, they're like little platforms that enable the bacteria to hang out and stay in one spot, despite the fact that they're constantly being, they're adjacent to this flowing tube of stuff.
Right. Yes. And these are really dynamic biofilms. I mean, they shed off and regrow constantly. And if you look at the fecal material, right place, right time, you can see beautiful sections of biofilms. There's a paper published just looking at biofilms that are on food material that's coming out in the fecal stream. It's a really very, very elegant system. And if our immune system helps these bacteria form the biofilm, it gives those bacteria a huge advantage over some other bacteria coming in that's trying to make its own biofilm and do its own thing.
Right. Okay. Yeah. So the immune system can selectively help certain bacteria form a biofilm to keep them there and thereby keep out other bacteria that might otherwise try and make their own biofilm and displace them.
Yeah. Basically, if you're another bacteria and you want to make your own biofilm, you almost have no chance, right? Because you've got to produce all these molecules all by yourself. So you may as well just go along with the host, use the host resource